when we're looking at these cells, and I'm going to go quickly sketch these out. You have three cells, so that one cell looks like that, there's another cell, looks like this, another cell. Looks like that. That's one condition. Another cell, a normal cell, thank you very much. Another cell condition, you have cells that look like this, three cells. And then the last condition, you have a cell look like this. And even some might be so these are all of these, and they labeled it for you. They're all animal cells. I mean, you already have it drawn in your notes. I don't care. You have it. I would draw in your notes. This is already drawn in your work. This was something we did yesterday. You had time in class to do it. Should have had, bless you, you should have had it done overnight. That was your objective. That was your homework last night. But I'm moving on either way. So in one situation, in this one, you see in the drawing that you have a big arrow. Looks like this. And you have a small arrow that looks like this. Now, why is there an arrow going in and an arrow going out? Why is that? Yeah.
Because that is the water going in and the water going out.
That's right. That's water going in, water going out. How do you know it's water? Because what else is at the top of this diagram that I did not put? Osmosis. They tell you it's osmosis, and they put in plants and animals. All right, so that's it. They tell you already, in animal cells you have two arrows. Why are the arrows going in and out? What is always happening when you're talking about diffusion or osmosis? Osmosis being the diffusion of water. What's always happening? Always moving. So always stuff going in and out, correct. When you have one arrow that's bigger than the other, what does this big arrow show you? What kind of movement? In this case, out, but it's what? The net movement is the movement of, after you subtract and add everything, the net movement is the one that's bigger. So most of the net, or most of the movements, which way? Out. You do have some going in, but most of it's going out. So over time, the inside of the cell is losing water, correct? So you get what? What does this look like? Shrinking. The cell's shrinking. That's why it looks like this. Okay. So then this cell here, in this condition, you see that the arrows are about the same. I'm not able to draw them very well, but they look about the same. And obviously it's happening in all three cells in each condition. Then in this one, your net movement, again, after you add and subtract everything, your net movement is in which direction? In. Because most of the movement is in. Is that good? Everybody understand that? Over time, what happens to the cell if it's under this condition, if it's under C, condition C, what it's going to get bigger. It's filling up. And yes, that's right. It might even burst because it might fill up so much it'll burst. Everybody okay with that? It's all making sense to you? All right. We call this lysing. It's called lysed in animal cells. Bless you. Lysed. We call this obviously the normal. And this one we call it crenated. Everybody okay with that so far? Yes or no? All right. So the net movement here is into or out of the cell. So what is this extracellular fluid? What's this fluid outside? Is it higher or lower in solute? Higher in solute, because water flows to where the solute's the highest. And since it's higher in solute, we call it hypertonic. Now, under normal conditions, what would it be considered? That's when they're what? Is the equal. The inside and the outside have equal concentrations of solute. We call that isotonic. Is everybody okay with that? Someone want to close that door because it's really annoying to hear all that mess out there. And C, what is it? Hypo. What does hypo mean? Why is the water going in? Where's the solute the highest? Inside. That's right. Everybody seems to get it here today. Where the water's going to move to where the water concentration is the highest, or the salt concentration, the solute concentration is the highest. The solute concentration is highest inside. So what is outside? Lower solute. So we call it hypo. Hypo means lower. Everybody okay with that? So that's it. That's all there is to it. You should be able to answer all the questions then based on that. As far as animals are concerned, all of that will be on the test, so we should be ready to go for that. Now there's three more situations that we're going to talk about, and that's in plants, right? Now plants have cell walls. So I'm going to draw it differently. I'm going to draw it with like 3 rectangles. And they round off the corners in the drawing, but I don't really care about that. I don't think you do either. Then I'm going to go ahead and use different colors. I'll say let's use green for the cell membrane. So I'm going to draw the cell membrane here. And the cell membrane over here is going to be filling up the entire space, pretty much. That's what I mean to do. If I mess up, it should be filling up the entire space. And then here as well, I'll make sure to fill up the entire space. So the black line is a cell wall. The green line is what? Cell membrane, correct. It's always inside the cell wall. And then there is within that obviously you have your nucleus, right? Your nucleolus, your Golgi body, all that. Everybody okay with that? This is all the nucleus. All of them have a nucleus, nucleolus, and all kinds of organelles, right? Chloroplasts, all that. But there is a bigger structure that looks something like this. And then over here, it takes up a larger spot and takes up the most here. It's the biggest here. We call this thing here, it's called the central vacuole. Central vacuole. All right. Now, in the first condition, we're going to label them 3 different letters. This is D, this is E, this is F. D, E, and F. In the first condition of the central vacuole, water is leaving in this direction. Of course, it's osmosis, so you're going to have water moving in both directions. But the net movement is out. So under what situations is the net movement out? When the outside, this is obviously a plant. I hope you know this is a plant. In what situation do you have water going out? What is it? Water going out when is the solute concentration higher or lower out here?
Higher.
Higher out here, so what condition is hypertonic is correct. In the second E situation, your in E, what is what situation are we dealing with there?
Isotonic.
Isotonic is correct. Why? Because the water moving in both directions is the same. Water moving in is equal to the water moving out. Now, they're going to be able to show you that in more than one way. They'll be able to show you that with big arrow, small arrow. They might give you numbers. They could give you percentages. They could tell you the solute concentration, ask you which directions of water moving. They could do a lot of different things. But, you know, in the case of the drawing, they chose to go ahead and use the arrows. And in this one, the net movement, the bigger arrow, the majority of the movement of water is into the cell. Does everybody agree with that? All right. So net movement's in. Of course, that's F. So again, the middle one, E, is called what? Isotonic. And F is hypotonic. Very good. It sounds like you guys studied well. So everybody's okay with that? All right. So this central vacuole, what's it doing? What's the central vacuole doing?
It's holding the water.
It's holding the water. So the central vacuole is actually the place where a plant cell stores its water. So when you're in a hypertonic solution and versus an isotonic solution, now there's water in the space in between in the cytoplasm as well, but any extra water gets put into this central vacuole. Are we good with this idea? So when you're looking at this, what you're looking at is as this central vacuole is emptied for whatever reason, in this case because it's hypertonic or salty out here or sugary or whatever, there's too much solute out here, water leaves, there's less pressure pushing up against this cell wall, correct? Yeah. The cell wall becomes flaccid. It's not as turgid, not as turgid. The pressure of this membrane up against the cell wall, that pressure, let me go ahead and draw it in red. This pressure here, this pressure, or lack thereof, pushing up against the cell wall, we call that Tuger pressure. Tugor pressure. The pressure pushing up against the cell wall. And of course, which do you think is higher? Is this, where's the Tuger pressure the highest?
In the model app.
In model F, so this is the highest Tuger pressure. In fact, we call it turgid. Stiff. It's stiff. Why? Because there's pressure pushing up against the cell wall. Why is there pressure pushing up against the cell wall here? Because the central vacuole is filled up, right? It's like filling up a tire, right? This is when as if your tire is deflated, this is when you inflate it. This is all filled up, there's a lot of pressure, right? This is when it's evacuated and you don't have as much pressure. When a plant is turgid, a plant is standing straight up. When a plant loses turgidity, loses that fluid in a hypertonic solution, it starts to wilt. Why is it wilting? Because it's losing that pressure, that pressure up against that's pushing up against the cell wall. Are we all good with this idea? All right. And of course, there's there's pressure on all three of them. It's just the least pressure is over here. Least turgid and most turgid. Agreed? All right. And by the way, when the cell has lost that turgidity, at least amount of turgidity, we call it plasmalized. All right. Where it's called, in animals, it's called crenated, right? It's called plasmalized in plants. Where it's a cell lyses in animal cells, they'll rupture. Here, a plant cell doesn't do that because it has a cell wall. Are we all good with that? So those are the two conditions that you really should know, right? Shouldn't have too much trouble taking a test on that on Thursday, correct? So those are your 6 conditions. Now let's go on and answer some questions and then move on. For each question, use A through F model 2. Which cells show a net increase in water? The cells showing a net increase in water. A, B, C, D, or F? C and F. Net increase in water. Very good. Net decrease in water? A and D. Net no change? B and E. So that's easy, yeah? Now, that's easy, but what that means is that on a test, you'd have to be able to say, well, hypertonic means it's going to lose water, hypotonic means it's going to gain water, isotonic means there's no net change. You have to be able to answer those questions that way, too. So just because I give it to you in this direction as a question in this direction doesn't mean that on Thursday you'll get a question in this direction. It might ask you the opposite, right? I'm giving you where the water's moving, and I'm asking you where was it hypertonic, hypotonic, or isotonic. I might give you that it's isotonic and ask you which direction does the water move. I might give you percentages, right? I might say it's 50% outside, 20% inside, right? And tell you is it hypertonic, isotonic, or hypotonic, right? So you have to be able to answer the question no matter what way it comes at you. All right. Consider the definition for osmosis and the net movement of water from dilute solution, high concentration of water to a concentrated solution. Describe the concentration of solution surrounding cells A and D, the extracellular. I want to know about the extracellular fluid for A and D. A and D. What are we going to say about A and D? Is the extracellular fluid is what? Is what? It is hypertonic, but what's the word? More or less concentrated?
It's more concentrated on the outside.
It's more concentrated in the extracellular fluid than the outside. Correct. Hypertonic means, that's what it means, right? Mm-hmm. More concentration of salt outside. Extracellular. What's the key word there? Extracellular. Extra meaning outside. And what's the other one? Intercellular, which means inside. Are we all good with this? All right. All right. Previous question, develop definitions for the following words. Hypertonic, hypotonic, isotonic. How do you define these? In terms of the words we just used. Hypertonic, the salt concentrate, extracellular fluid is higher. Hypotonic, the extracellular fluid is lower. Concentration of what? Solute, correct. And isotonic, the extracellular fluid, is the same concentration inside and out. All right, stop and consider hypertonic solutions. Describe what has happened to the plant. Well, this one, In this one, you have highest turgidity, the plant is standing straight up, it's happy, right? That's when you have a lot of water, very little salt outside. In this one, in the hypertonic solution, they want to know the hypertonic solution first. The plant is become less turgid. The plant is becoming wilting. The plant is wilting. The plant is dying. It's plasmolized. The animal cell is crenated. It's dying. It's shrinking. Then both the cells are shrinking. Yes? Mm-hmm. Water's leaving, the cell's shrinking. What word would be used if the cell were from an animal? We already said that. Let's move on. Consider the cells of Model 2 that are hypertonic solutions. Describe the changes in the plant cell. Model 2, hypotonic solution. What's this changes in the plant cell?
Yeah, it's a lot of water.
A lot of water inside. The cells become bigger. It's swollen. It's pushing up against the cell wall more. So it's what? Highest turgidity. It's stiffer. It's standing straight up. So when animal cells are in a hypotonic solution, they can undergo lysis. However, plant cells do not. They only become turgid. Why don't they? Yeah.
Because they have a cell wall.
They have a cell wall that stops them from bursting. Okay, now let's go on and talk about the extension questions. Let's apply these principles to something. Tomorrow, there will be a 10-question test, so there will be no notes. Okay, then on Thursday, there'll be a 10-question test. I recorded it so that there's no question. All right. So you can either listen or not. It's up to you. Or you keep talking. Go ahead. All right, losses. All right, so obviously you have water on this side, mostly water. You have solute on this side. So you should be able to answer, describe the two properties. Exchange the surface would facilitate rapid diffusion across rapid diffusion rates, right? When they say exchange surface, what do they mean? What properties of this surface would facilitate rapid diffusion? So, large pores.
What page is this?
All right, you can figure it out. I'm confident you have the ability.
I appreciate that.
So large pores, so permeable to water and permeable to the solute, right? So large pores and permeable to the solute. Okay, that's reasonable. Describe two biologically important features of diffusion. What are important? What's important about diffusion? One, they do what? Go from high to low. And two, what's another thing about diffusion? Reaches dynamic equilibrium. There's a lot of answers you can put in here, but certainly that molecules will move from high concentration, low concentration. Secondly, you're going to reach that dynamic equilibrium. At some point, they're going to be equal. So in other words, high to low, but they are moving in both directions. So they're moving in both directions. You're going to get movement of molecules in both directions. It's just that most of the movement's towards from high to low, and secondly, there will be a point where the both sides will be about equal concentration. So the movement in both directions will mean that there won't be any change at all in the concentration. Is that correct? Everybody okay with that? We've gone over that. So is this is not new, right? Does anybody think that they don't understand anything here?
I mean, a little bit, not really.
All right, describe how facilitated fusion is achieved for small polar ions. So for small polar ions, I'm not even going to go and describe. You can go back on the previous page and describe it. The previous page, if you flip, you'll see that everything's described clearly, step by step. You can answer that. And for glucose, we went over and described glucose as a carrier, right? So small polar ions, I can tell you it's probably going to be a channel. And larger molecules that are polar, you're probably talking about a carrier of some kind. Correct?
Yeah.
All right. And you guys can look in. It wants a description, so you need to describe in detail. How concentration gradients are maintained across membranes. Now, this is going to be tough. This is where we start to get into something else. Something else is going on. So, often in biological systems, and you're going to see that in mitochondria and in chloroplasts, but you're going to see it in a lot of different places. You're going to have a membrane, and on one side of the membrane, you're going to have a high. And that high can be anything. Could be sodium, but you could be high sodium on one side. And you can have low sodium on the other. And you know what? Let me use a better example. You have a high hydrogen on one side and a low hydrogen ion concentration on the other. Is that clear? So where would it normally flow if it could? If you just had a channel, if you just put a channel in, what would happen? What would happen if I just had this little channel? It's an ion, so it needs a channel, right? We know this ion has the channel has a nonpolar end, it has polar sides, correct? Yes. We've drawn this like 50 times, so everybody should know that. So now this hydrogen ion is going to go from high to what? Low. To low. Eventually, what's going to happen? Eventually, you're going to get, and you're always getting movement. It's a channel, so you're always getting movement in both directions, correct? But you have more movement, bigger arrow in this direction than you do in this one, because it's high on this side and low on that side, right? Eventually, what are you going to reach? The equilibrium, correct. At some point, you're going to reach this thing that we call equilibrium, this dynamic equilibrium. So if there's hydrogen ions over here, there's 2 hydrogen ions over there, there's going to be 2 over here, right? And you're still going to get movement. Let me be clear. You're still going to have movement in both directions. It's just that they're going to be equal. For every 1 hydrogen ion that goes over there, there'll be 1 hydrogen ion that comes back in. So there's never going to be a change. Is that clear? All right. So that's no good for you, because it has to do with batteries. Anybody know what a battery is? Yes. Do you? Let's test it. Let's test that.
You thought you knew what a battery is?
So you do. Okay, so there's this. This is the positive side. This is the negative side. There's a little divider. There's a chemical inside. There's usually a divider inside the battery. Almost in alkali batteries, lithium-ion batteries pretty much run the same idea. Most batteries are going to work this way. You got a separation of charges, positives and negatives, and then you put a conductor on, and that allows. So they usually a conductor's made of copper. And you connect the 2 sides, and you can connect it with, wow, I don't even know where to begin with you two. I hope your mother listens to this, though. But we could put like we could put a light bulb in here. And, of course, you put a light bulb in here, it's going to light up as long as the electrons are flowing, and the electrons are going to flow from the negative towards the positive because opposites attract, right? Yes. We call that a current. Everybody covered that in middle school. You might cover it in conceptual physics. All right. Oh, I'm so glad that you know this so well. All right. So your body works the same way. You need, what happens when you run out of electrons? The battery dies, and so what? The light bulb goes off. Do you want your brain to go off? So we better make our batteries last. Let's take a look at our batteries. What we have is our batteries are separated. Do you see this separator right here? Let me pause it. So the water, here we have, as we said, that once all the electrons leave and move over here, the battery dies. There's no more electricity because all the electrons have moved. Correct? So that means no more light. Well, this is a battery too, isn't it? So if I draw a membrane, and on this side I have 100 positives, and then I draw a little channel, and then I have only 2 positives on this side. This movement of ions from this side from high to low is current. That's energy. Just like the battery was energy. So your cell membranes act like a separator, and the charged ions across the membrane actually are what you call a nerve signal. That's what allows for nerves to get sent from one end of a cell to the next.
So you know how like the doctor I put a hammer to your knee and I pop up.
Everything, the thought that you just had, that memory, that idea, that the love, the hate, it's all about all these nerve signals, all these what we call depolarization and repolarization of your neurons. So it's across the cell membrane, you see? And you have a separation of charges. Look, hear me. Separation of charges. As soon as you separate charges, you have something that I know you've all heard of before. Potential energy. That's right. But as soon as you connect them and you allow the flow to happen, that's kinetic energy. That's movement. So before that, you have potential energy. So you have electrical potential energy. You have a battery. But if you reach equilibrium, if you allow this to go to equilibrium, hear me clearly. If you allow these 100 charges to go across the membrane and come to dynamic equilibrium, then you've run out of charges. There's no more energy left. You have zero potential energy left once you've reached equilibrium. Is that clear? That equilibrium will occur if you let the charges flow. You need the charges to flow in order to get electricity, right? To get kinetic energy. You need to have some of it go. How do you keep it, though, from going to zero? Yeah.
So the people that are, I don't know what's the other word for this, like sort of like not really like vegetables.
Yeah.
Like brain dead. That's would that work with them or no?
Depends on the cause. Again, like disease. Remember we went over disease. It depends on the cause of the problem. So the problem is they're not responsive. But why are they not responsive? Are the cells dead? Do they have a problem with connecting? Some people are what's called trapped syndrome. I forget what it's called, but something syndrome where you're thinking, you're able to observe, but you can't act. Some people don't have any brain function at all. So it all depends on why they're in this situation they're in. Yes?
So to keep like the cells growing, you gotta eat healthy and stuff?
No, listen. You're almost there. Listen. So you have high on one side, low on the other. As soon as you open a channel, you have movement of ions. That's electricity. You got a thought. There's your thought. But you need to do what? What happens when you run out of the battery? How do you get more energy in the battery? What do you do with your cell phones? You charge it. So you got to charge this. You got to move those ions back. But think about what that means. If you had 100, moves over to 2, then some time goes on, and I'm going to go ahead and just use a different color to separate here. And some time goes on, and now you got 75 is moving to 28, and then a little more time, 50 goes down to 52, all right? So now what? It's getting closer and closer. Now we're near dynamic equilibrium. Now we're at 0. This is no good. How do we stop this? We have to do what? We have to reverse it. We have to reverse the flow. But we just said that things only go from high to low with no extra energy. What do we need to do? We have to add energy. We have to recharge the system. By the way we do it is we add something called ATP. And we get ATP from only one, really, well, any macromolecule, but really the one we talk about is glucose.
Yeah.
Glucose goes ATP, then we can use the ATP to move things from low to high.
Yeah, man.
Now we can recharge that membrane. So yeah, we want it to come down. We want to discharge it. It's called discharging. But after we've discharged it, we want to do what? Recharge it. And we use food. We use food to do that. That's why you eat. It's the only reason you breathe oxygen. Is just to recharge your membranes so that you can keep working. Your cells can recharge the high and lows, reinstate the concentration gradients. If you don't have a concentration gradient, you got problems for various reasons. So this particular mechanism we just described happens in mitochondria. So this is also, there's a word for this type of transport. It's called active transport. Active transport is when things go from low to high. How many different kinds of passive transport do you know? Passive means what? No extra energy. How many different kinds of passive transports are you aware of? How many different kinds of passive transports are you aware of? How many? I want you to move. If I have to move you, you'll both move. So, transport. How many different kinds of passive transports do you know?
Passive transport.
Passive transports. I guess none. From what I'm hearing is none.
Hold on, man.
All right, could you two please move? You, dear, get up and move over there in that table next to Mr. Shorts. You, dear, move up and sit next to Tariq. Thank you very much. Do it now, or one of you or both of you are going to the office. So let's do that now. Thank you. So, passive transport. What two kinds of passive transport do you have? What have you been studying all week? Osmosis and diffusion.
Did they say that?
I didn't hear it. No one spoke. So osmosis is the passive transport diffusion of water. Passive meaning high to low. What kinds of diffusion have we talked about? What different kinds of passive diffusion have we talked about? Flip the page back. Since those of you are not studying, not reading your notes every night. What does the top of that page say? Passive transport. What kinds of passive transport do you have? Simple diffusion. Facilitate diffusion. Facilitate diffusion with carriers. Facilitate diffusion with channels. And osmosis. So it sounds like when you get your test on Thursday, you know what it's going to be on. The 4 or 5 different kinds of passive transport. What are they? How do they work? They're going from high to low. And then active transport. Active transport is going from low to high, and you're using ATP. These are your permanent seats, ladies. All right? Thank you. Are we all clear on this?
Yes, sir.
So, does anybody have any questions about passive or active transport? All right. I think it's time to go, right? When is this class over? You got 1 minute, yeah? Whose phone is this?
Patient B.
Whose phone is this? This is going to the office. You can talk to them in the office about it. Thank your friends for that. I warned them 3 times. I said, stop, stop, stop. What do you want me to do?
I heard you.
What do you want me to do? I'm not gonna sit here.
He didn't say stop, he said put the phone down.
I said it several times. I don't know what's wrong with me. I think I'm just gonna sit there and listen to that nonsense. Got nothing coming. I think I ain't doing it.
He just said everybody who
Picture
got
me.
phones, I'll put them
I'm
up.
sitting, bro.
He said out loud. That's what he said.
Over a mic, over a speaker.
And it go through the whole
But they were
class.
too busy playing and talking.
That video's not that serious.
It is serious. They're not gonna do well on your tests because they're not listening, not paying attention.
I stopped the video. I just don't use it.
I usually check the time.
Yeah, you're all done. I haven't put it in.
Can we come during lunch? Because I tried to come this morning.
Yeah, I was here, but I was in the other room. During lunch, you for what?
Because I need to turn in my study guide and my notes from Khan Academy from last quarter.
Put your name on a slip of paper. I'll turn it in the office. You can see them after school. I don't know what they're gonna do with it though. It's up to them. Why'd you give it to them?
It was just out. She just took it. I didn't know she had it.
Well, you can have that discussion with the office though. I don't know what else. I tell you girls, I said, you guys gotta stop. You guys gotta stop. You guys gotta stop. And then I'm supposed to do what? I can't
But
just,
I wasn't the one who was on it.
I know, I appreciate that. But I'm sitting there telling you repeatedly, something's gonna happen. You need to stop. You need to stop. What I would do if I were you, I would have gotten, spell name.
A-N-D-R-E.
Say again?
A-N-D-R-E.
A-N-D-R-E. Uh-huh.
U-P-T-O-N.
15. 10. 10 or 9, I don't remember. Lunch. I mean, you're putting me in a real tough spot here. I'm talking to the class. Look at all these handouts. Could you go around and collect these for me before you leave? Look at all these notes, especially back there at that table, the interesting table I have. Is this your work? Mm-mm. Look at this. The 2 kids that need it the most are the ones that leave it behind. That's all right. They're not getting another copy. They could fail. I don't care. I'm sick of this nonsense. Well, I feel bad for you. I really do. Look at this. Unbelievable. 3 of them. I'm gonna make sure I'm gonna wait till they come and ask me for it. Yeah, throw the rest of those away so nobody gets it.
You're supposed to write my name or something.
All right, so I moved them, yes?
Yeah. Wait, also you didn't put him in Khan Academy, so that's missing, but I turned it in. That's another thing. So.
Well, you can see me at lunch or after school for that, but you're, do you realize you're wrong for having the cell phone out? Why did they have your cell phone?
Because it was out.
And then they took it? They were watching it, they were laughing about it, so were they on your phone during class, playing around with your cell phone? Why'd you let that happen?
Well, I was paying attention to the notes. I wasn't paying attention to my phone most of the class.
And they just took it without your permission?
They always do that. Like, I don't care. I mean...
I understand. You're their friend, so you don't really care if they play around on your phone.
Yeah, it would be different if I didn't know them,
But I understand. You know them. I'm not trying to blame you for anything here. I just want to understand the situation. Is that clear?
Yeah.
So you have your phone out, and then your friends take it, and they're your friends, so you're not gonna tell them no. Okay, I get that. So let me just document the whole thing, and then just give you your phone back.
Okay.
'Cause I don't know why. I understand your situation. You're in a tough situation. You don't want to cause trouble to your friends. I get it. I've been there. I've done stupid things myself. All right. Give me the names again.
Of them,
You and them.
Stevenson,
Stevenson,
Harris,
Harris, and Kelsey, and then Henderson. Yeah. And then moderate. Cell phone. Sydney. Was had her phone Harrison Anderson. Yes. Harrison Anderson. During class. Laughing and playing on the phone while instruction is going on without your knowledge, yes?
I mean, I guess I knew that they had it, but I wasn't paying attention to that. I didn't care.
So that's it needs knowledge. She paying attention to her work. I warned Anderson and Harris several times, and finally to take the phone away and move their seats. Sad that I have that. I really don't like acting like a middle school. I don't mind doing this if I'm in middle school, but to do this and have to deal with this in a high school, it's just sad. Here you go, dear.
Thanks.
Let me write you a pass. Where are you going?
Simpson.
Also, get in dress code. Don't leave here. Go to Miss Simpson's class. Dress code. Right now. I'm walking over there right now. Use our machines to see this depolarization, the movement of ions across the membrane. This is what it looks like. Are you ready? We have machines that can read them. You tell me if you've seen this before. Ready? Bleep.
Oh, yes.
Bleep. Bleep. Right? What happens when there's no more bleeping? And what do we call that? This area here, this bleeping, is the electrical signals of your body releasing the ions and then repolarizing, doing active transport to recreate the concentration gradient, and then releasing them, and then recreating them. So what you're getting is release, recreate, release, recreate, release, recreate. When the minute that stops, we're done. Right? So remember that. When you see it in the movies, when you see it in the hospitals, that's what you're recording. Whether it be your brain or your heart or your body, all these things are doing the same thing.
What if you eat too much?
You turn into fat, and you put it away in your body for storage later. Say what now?